So thank you all for attending. We are really excited for this opportunity to showcase all of the really amazing work that students were able to accomplish. And we have 5 really amazing presenters today that are joining us. All presentations will be judged by me, and a recording will be passed to Alex Parola, who is at Princeton University as a graduate student, and he is in their mechanical and aerospace engineering department. So those will be the judges for this session. And again, this is a full house, so it's really important that we stick to that 8 minutes for presentation and then 2 minutes for questions. At the end of that 10 minutes total, I will move on to the next presenter just to make sure we're being mindful of everybody's time and that they have enough time to present their work. And awesome. So I would love to get started with Sabrina. And so I am going to ask her right now to join us. Hi, Sabrina.
Hi.
And so I can hear you, I can see you, and if you click that share screen button now, you'll be able to share. And just for context for all student presenters, it's best if you share like a particular tab if you're presenting from a browser rather than sharing your whole screen. It'll make things a little smoother. All right, take it away.
Awesome. Can everyone see my screen?
I cannot see it just yet. Click it should look like a monitor towards the bottom panel where there's like the mute and the video button.
Hold on. Okay, can you see it now?
Yes, it's loading.
Okay.
Yep, all good.
Okay, cool. Hi everyone, my name is Sabrina Toor. Just a little bit about me. I'm a senior at Rock Canyon High School. I'm the founder of the Society of Women Engineers at my school. I'm the vice president of Med Club at my school as well. And I'm also interested in pursuing a career in medicine. So I researched the history and effectiveness of immunotherapy in cancer treatment. Here's just a brief overview of the topics that I will be covering today. And then here's a timeline of some cancer-related events. One particular event that I would like to point out is in 1967, where Jacques Miller confirmed the existence of T cells and found that that they all develop in the thymus gland. This is important because it's contributed to the modern forms of immunotherapy that we have today. So cancer is generally divided into solid tumors and blood cancers. Solid tumors have the ability to form a physical mass, while blood cancers are just an overgrowth of white blood cells. The 2 main types of blood cancers are lymphoma and leukemia. Leukemias typically arise in the lymphocytes, while leukemias arise in the leukocytes. So here's the main types of cancer treatments that are currently used. Chemotherapy, surgery, and radiation therapy are definitely the most common. So as you can see, each cancer treatment does have a significant number of side effects that comes along with it. Chemotherapy and radiation are specifically harmful because of the damage that it can do to DNA of cells. It's beneficial when it damages the DNA of cancer cells, but it can also damage the DNA of healthy cells, which can lead to hair loss and fatigue and other unwanted side effects. So the immune system is basically a network of cells and proteins that work together to recognize, remember, and attack pathogens. Immunotherapy is a treatment that utilizes the body's natural defenses to attack these cancer cells rather than just attacking any general pathogens that enter the body. So there are multiple types of immunotherapies, but my paper focuses on adoptive cellular therapy specifically. There are 4 main types, including TCR therapy, TIL therapy, and NK cell therapy. The focus of my paper will be chimeric antigen receptor therapy, or CAR T-cell therapy. So CAR T cells have the ability to bind to and attack cancer cells, even if they lack antigens or an MHC on their surface. The MHC is a major histocompatibility complex, and this basically just allows any immune cells to bind to cancer cells But because CAR T cells have the additional receptor on them, they don't need an antigen or MHC, making them more efficient and effective at targeting cancer cells. So there's some key factors to understanding the effectiveness of immunotherapy. Tumors can either be hot or cold, and their microenvironments can either be fibrotic or vascular. A fibrotic cold tumor will typically be harder to treat with immunotherapy just because it lacks blood vessels and it lacks immune cells, while vascular hot tumors will be easier to treat with immunotherapy due to the presence of blood cells as well as the presence of activated immune cells, meaning that the immune system has recognized the cancer. So there are 4 main phases to clinical trials. Each one has an increasing number of participants in it. Phase 3, after phase 3, a pharmaceutical company can submit their drug to the FDA in order to be approved. And once it is approved, it moves on to phase 4, where the long-term side effects are monitored. So here's a map of some global clinical trials of CAR T-cell therapy specifically around the world. As you can see, the United States, Europe, and East Asia has the most presence of clinical trials in CAR T-cell therapy. Currently, since 2017, there have been 4 FDA-approved CAR T-cell therapies. Kymriah was the first of its kind, and all of these have been approved for either lymphoma or leukemia. So I analyzed 4 main clinical trials in my paper, and this is just a table summarizing each one of those. A specific term that I would like to identify is complete remission, and this basically means that there are no detectable traces of cancer left, and the patients don't experience any symptoms of cancer anymore. This is the scale that I'll be using to evaluate the effectiveness of CAR T-cell therapy, which I'll talk about more in the next slide. So here's a graph talking specifically about the percentages of complete remission. As you can see, clinical trial 1 has had the highest percent of complete remission at 40%. The average complete remission across all 4 clinical trials is 26.5%, which is relatively high compared to other forms of cancer treatments. So some pros and cons of CAR T-cell therapy. Some pros are that it doesn't require antigens or an MHC, as I spoke about earlier. This just makes it easier to target any type of cancer cell, specifically lymphoma or leukemia, that is present in the body. It also has long-lasting effects compared to natural killer therapy, which only lasts about 2 weeks in the body. CAR T-cell therapy actually is designed to last for a lifetime. So similar to how your immune system can recognize certain viruses, CAR T-cell therapy is designed to do the same thing, but for cancer cells. It can also treat both leukemia and lymphoma, and it can do this because each type of cancer only occurs in a certain type of white blood cell. So because of this, there's only one factor contributing to its growth, and it's easy to target. Of course, there are some cons. This would include cytokine release syndrome, which is basically when your immune system overreacts to certain cancer cells, which can cause inflammation, low blood pressure, and neurotoxicity. But if these are addressed early on, they don't become too harmful. Another con would be that it still needs to be studied in solid tumors more. So solid tumors have multiple factors that contribute to their growth. And because of this, it's difficult for CAR T-cell therapy to target only one of these factors. Another con would actually be the cost, because it can cost up to $400,000, while chemotherapy costs up to $12,000, just for a comparison. So in conclusion, CAR T-cell therapy is highly effective in treating leukemias and lymphomas, and scientists are currently working towards creating a treatment for treating solid tumors with this therapy as well. And it's generally important because the current types of cancer treatments have a plethora of side effects that are related with them. Cytokine release syndrome is the only serious one that's related to CAR T-cell therapy, so it makes for a great alternative. I would like to thank my mentor, Jennifer, for helping me throughout this entire research process. It was amazing working with her. And then here is my contact information. I'm currently working towards publishing my research in a scientific journal article, but if you have any questions, please feel free to email me. Thank you.
Thank you so much, Sabrina. Great presentation. And we're right on time. So we have just about 2 minutes for questions for Sabrina. You can either put those in the chat, that's to the side, the chat or the Q&A. I'll check both. If you want to ask directly, you can also ask to share your screen and to ask a question directly to Sabrina. One question to get started is, I'm curious what was most shocking or surprising to you as you were diving into this area of research?
I think it was definitely like I didn't even hear about immunotherapy, so it was just working with my mentor Jennifer and like learning that immunotherapy existed, because I knew that the current forms of cancer treatment have such high side effects. So it was really crazy to see that there was this treatment that has less side effects. Of course, with a cytokine release syndrome being the only one, but it was really great to see that technologies come so far and that we have the ability to just work with the body's immune system and just make it target cancerous cells. So I thought that was pretty cool.
For sure. So I'd love to have some audience questions in the chat or from the Q&A. We'll give a few more seconds. So here we got one. Great talk, Sabrina from Selena. Can you tell us a bit about the differences in the 4 clinical trials you compared? Were they all using the same CARs?
Yeah, so they actually were. They used CAR T-cell 19. That was the specific type that they used. Each one analyzed something slightly different. So the 4th clinical trial was actually monitoring which dosage of CAR T-cell therapy would be the best in patients. And then clinical trials 1 and 2 were actually used in conjunction with chemotherapy. So that actually proves that chemotherapy along with CAR T-cell therapy is more effective than just chemotherapy, because the average of complete remission across all 4 clinical trials was 26.5%, but just using chemotherapy on its own only has a complete remission rate of 22.5%.
Great. If you have any other questions for Sabrina, feel free to chat with Sabrina in the chat after her talk. But for now, we're going to pull up Ekansh. Thank you so much, Sabrina.
Thank you.
Hi. And then Sabrina, I just encourage you to check the Q&A because another question came through. And hi, Ekansh, I can see you. Can you talk just so we make sure your audio is good? You're on mute.
Oh, sorry about that.
No worries. I can see your screen. No worries.
Okay. So, hi, my name is Ekansh Kushwaha, and for my project, I use liver single-cell RNA-seq data to find new gamma delta T cell subtypes, and I did all of this with the help of my mentor, Zachary. So first, to just establish a little bit of background, the body's immune system protects us from all sorts of pathogens, and those pathogens may include various diseases such as bacteria, viruses, and cancerous cells that may form in our body. And T cells are the primary mediators in the adaptive immune response, as these are the cells that attack and get rid of foreign substances in our body, such as bacteria, cancer cells, viruses, and parasites. So I talked about the adaptive immune response. There's actually 2 different types of responses in the immune system. There's the adaptive and the innate immune response. So the innate immune response is what you're born with, and it doesn't really remember any pathogens that have attacked your body in the past. And it's as you can see in this graph on the right, it's a pretty linear response. It attacks like all pathogens at the same amount. And that's where the adaptive immune response comes in, as that can basically dissect any antigens that infect our body and find out how to kill it in a much quicker way. But this does take a little bit of time. So yeah, the innate and adaptive immune response work hand in hand. So as you can see in the graph on the left, the innate immune response does like a bit of the work, and then the adaptive immune response and comes and it quickly ramps up and just like kills it really quick. And the neat thing about adaptive immune response is that they have memory cells, so they actually remember each antigen and how to kill it in the quickest way possible, which is really great because as you can see, the second time it was exposed to microbe X, it reacted much quicker and it killed it much faster. So this is great for immune cells. And the reason why I chose T cells is because I learned something really interesting about them while working with my mentor. And that is that like B and T cells can actually undergo a process called VDJ recombination, and that can change their DNA sequence. So this is great for immune cells as they, like I said earlier, they can adapt to any pathogens that might attack and kill them in the most efficient manner possible. And this also means that there are many different T cell subtypes that are possible. So this is like a great area to research. There's two main types of T cells. They're the alpha beta and the gamma delta T cells. The thing with the alpha beta T cells is that they've been thoroughly researched in the past, whereas gamma delta T cells have only recently gained attention in the scientific community due to their role in autoimmune diseases, various infections, and cancer. So as such, not much is known yet about T cells in general, and specifically like gamma delta T cells and their subtypes. So this was like a great place to do my research. And due to VDJ recombination, me and my mentor have calculated like through the different types of chains, the gamma and delta chains that are available. We've calculated that there are about 120 main gamma delta subtypes. And out of those, 2 have been thoroughly researched and about 10 more have been identified. So there's a lot of room for more research. So for my methods, we first collected liver single-cell RNA-seq data from the Pangalau, Human Cell Atlas, NCBI, and Single Cell Atlas datasets. And we got both healthy and diseased tissue so that there was like more of a variety. And I got 5 healthy data sets and 3 diseased data sets. And the disease that they had was hepatocellular carcinoma. This data was then imported into RStudio so that it could be processed through seurat. And seurat is an R library, and that is the easiest way to analyze single-cell RNA-seq data. So for my methods, I followed the guided clustering tutorial, and you can see my code on the left. The guided clustering tutorial from the Seurat website helped me get 2 of my main pipelines, which helped to normalize my data and find the variable features and scale it. And then it also helped me make the graph that I'll show a little later, which made it really easy to analyze my data. I also used functions to get rid of cells that were in between cell cycles, because when a cell is in between a cell cycle, it actually has a really high expression value in my data. So that can skew my results in downstream analysis. So I got rid of them here through a regression. So for my results, I used the graphing functions built into the Seurat package to make a graph of the different clusters. And the clusters are all color-coded for easier readability. I then made a feature plot, which would express the genes of interest that are in the different clusters. And since I was looking for gamma delta T cells, my genes of interest were TRDC, TRGC, and TRGC2, and CD3. The TRDC and the TRGC genes are specific to the gamma delta T cell cluster, which is why I picked them. And then I picked the CD3 genes because they are expressed in all immune cells, so that could help me narrow down my feature plots and which clusters I should subset out. So this is a UMAP on the left, which just shows all the different clusters. And then on the right, these are the feature plots of each of the different genes. So as you can see, there's expression in cluster 12, but not all of cluster 12. So I had to extract only the bottom part of cluster 12 out from my first data set. And then in my second data set, I had to go to cluster 2, and basically all of that has expression of these genes. So I just extracted that out immediately. Then for this one, I got all of cluster 12 since that expressed everything. And then so on with the rest. As you can see, in this one, it was cluster 14 that had a lot of expression. And then in this one, 3, 5, and 7 have a lot of expression. 3 does not express that much TRDC, but me and my mentor believe that's because it expresses a different TRDC gene, which I did not include when I ran my code. This one is the bottom of cluster 4. As you can see, all of the gamma delta T cell specific genes are expressed there. This one was cluster 6. This was actually a little bit tricky because they were scattered all over 1, 2, 3, and 6. But my mentor decided to be on the safe side and to reduce the amount of non-gamma delta T cells in my subset to only take 6 instead of subsetting everything out and identifying it. In this one, I got all of cluster 12, but I decided to further process this later to remove any alpha beta cells that might pop up there so that I can reduce variability. And then this is the final UMAP I made. This is what happened when I combined all of the different subsets. And to make the different subsets merge into this, I actually had to use a different library called Harmony. And I followed the Seurat version 3 Harmony Vignette as the foundation for my code. If I did not run Harmony, all of the different subsets would be scattered all over the place, and they wouldn't be nice and neat like this, so I can actually analyze it. So I'm not done with my project yet, but so far we have collected all of the single cell RNA data sets from the liver that I have to use. I have identified all of the gamma delta T cell and subsetted that out. And then I've also combined all of the subsets to make my final UMAP. And then in the next few weeks, me and my mentor will be using 2 different functions called GP Profiler and Find All Markers to hopefully identify a new gamma delta T cell subtype. Now you might be wondering why all of this is relevant. Well, a new gamma delta T cell subtype that I might find could be discovered to play an extremely important role in fighting a new viral disease or even cancer. And in the future, with improved technology, we could actually use CRISPR, a technology used to edit the gene sequence, and could convert gamma delta T cells or even alpha beta T cells into this specific type of gamma delta T cell, since I'll be getting the signature for it. So yeah, that could be really great to cure cancer or viral diseases really quickly. And I would like to thank my mentor, Zachary. He helped me a lot with this project, and I definitely could not have done this without him. Thank you. Oh, can you not see my screen?
Okay, it just faded out for me for a little bit, but then it came right back. So I just wanted to make sure that others could see it. I was able to catch it. But if anybody couldn't see it, just let us know. Great, thank you so much. Awesome job and awesome work. And it seems like you have really exciting things on the horizon as you wrap up your project, which is awesome. I put one quick question in the chat, and I would love for others to contribute in the Q&A or the chat, but I'm kind of interested in these B and T cells. You kind of highlighted that they're unique and that they can undergo recombination. Is that something that, do you know if that's unique to those cells in particular? And how does that compare to other cell types?
Yeah, so far with the research we have currently, B and T cells are the only types that can have VDJ recombination. Yeah, other cells, they just use the gene that they were made with, I guess, and then they just express those using RNA polymerase normal. But these can actually change their own DNA, and normally you'd have to use CRISPR or something to actually cut and paste different parts. So that was really fascinating for me to learn about.
Yeah, that's really wild. Any other questions in the chat or the Q&A? Here's one. What programming language and software have you used so far?
So for this project, I had to use R, and I used the R Studio IDE. And I just installed the library Seurat to run all of this. And I needed a few others, but that was for some weird data sets that I had.
Awesome. And if you want, like afterwards, you can put those in the chat too, just so everybody knows, in case this person's wondering. Great, awesome job. I am going to move us on to our next presenter. Again, if you have continuing questions, feel free to put them in the Q&A or the chat, and Ekansh could look through them as he's in the room still. And now I'm going to pull up Khushi to share. Thanks so much. Hi.
Hi, sorry, give me 1 minute.
Yep, no worries.
Can you see my screen, or is it?
I do. I see that you're sharing currently right now the same screen. Yep. And now I see your PowerPoint, your Google Slides.
Is it?
I don't see the speaker notes that you opened. Nope. All I see is your slide so far.
Okay, cool. So I'll just get started then.
Yeah, perfect.
Hi, my name is Khushi, and my mentor is Christine Yoon, and my project is optimizing small molecule inhibition of the sJIA and mass biomarker IL-18 via Autodock Vina and Swiss ADME. So what is sJIA? Juvenile idiopathic arthritis, or JIA, is a category of autoimmune conditions that affects about 300,000 children in the United States. Systemic juvenile idiopathic arthritis, or sJIA, is a subtype of JIA and affects approximately 10 to 20% of patients with JIA. Characterized by the systemic inflammation of joints and corresponding joint pain, sJIA is a condition in which the hyperactive innate immune system attacks one's own joint cells and organ cells. In addition to joint inflammation, common symptoms include fever, rash, and swelling of lymph nodes. Guidelines for the formal classification of systemic arthritis by the International League of Associations for Rheumatology involve at least 2-week-long arthritis and fever, as well as one of other common symptoms, such as arthritis rash. To be diagnosed with sJIA in particular to achieve the juvenile term, a patient must be at or under the age of 16 at onset. sJIA has no known cause, as its name idiopathic suggests, but it is speculated to be influenced by a combination of genetic and environmental factors. While the cause of sJIA is unknown, there are known biomarkers, notably high concentrations of certain inflammatory cytokines, which are biological signaling molecules in the immune system. These inflammatory cytokines pertaining to sJIA are interleukin-1 or IL-1, interleukin-6 or IL-6, and interleukin-18 or IL-18, among others. Current treatment involves IL-1 and IL-6 receptor blockade, which means that treatment inhibits the signaling pathway of these interleukins by preventing the interleukins from binding to their respective receptors. There are no current treatments for sJIA via IL-18, but IL-18 receptor blockade and IL-18 blockade, so directly turning IL-18 inactive by binding something to it, both serve as potential treatments for sJIA patients non-responsive to prior treatment, or sJIA patients with a history of macrophage activation syndrome, which I'll touch more on later. Interleukin-18 is a pro-inflammatory cytokine that belongs to the IL-1 cytokine family. So active IL-18 binds to its alpha receptor IL-18R-alpha, which is shown in the diagram to the right, and it binds with IL-18R-alpha with low affinity, and then binds to its co-receptor IL-18R-beta, forming a high affinity complex. This induces the formation of a complex with the myeloid differentiating factor, or MYD88, the IL-1 receptor-associated kinase, or IRAK, and the TRAF6 protein. The formation of this other complex then activates two pathways involved in prolonging inflammation. The first is the NF-κ-beta pathway. This induces the transcription of inflammatory genes, like those of other pro-inflammatory cytokines besides IL-18, among other genes involved in inflammation. The second pathway that is activated is the mitogen-activated protein kinase, or MAPK pathway. This is involved in further IL-18 production. IL-18's responsibility for the downstream activation of other pro-inflammatory cytokines and further activation of more IL-18 molecules shows its critical role in inflammatory responses. Thus, high concentrations of IL-18 can influence autoimmune and autoinflammatory disorders. In the context of sJIA, IL-18's free concentration is elevated, and even more elevated in sJIA patients who have developed macrophage activation syndrome, or MAS, in the past. It's a condition sJIA patients are at a higher risk for, in which there is an overproduction of T cells and correspondingly macrophages, so these are immune system defense cells. And when these immune system defense cells are produced, this induces the production of inflammatory cytokines on a higher level than just in sJIA alone, and this can damage organs such as the heart and liver. Prior to research conducted by biochemists from the University of Texas Health Science Center and Oklahoma State University, there were no published small molecules inhibiting IL-18's bioactivity. These researchers proposed 3 small molecules as potential inhibitors of IL-18 being activated and then preventing IL-18 from binding to its receptors. The objective of this project is to use online molecular docking simulations to bind these proposed small molecules to IL-18, as well as to derive analogs of these molecules, which are essentially these molecules with additional atoms added to them, and bind these analogs to IL-18 to figure out how to optimize IL-18 inhibition. Today, I will be going into how I optimize the small molecule inhibition of IL-18 with one of these molecules proposed by the biochemists, NSC61610, and its analogs. So in order to do so, I used 4 main tools. The first is Autodock Vina. It's a molecular docking software that allows you to dock a molecule of your choice onto a target protein. It then calculates the binding energy of this interaction among other parameters. What you need to know is that the lower the binding energy calculated, the better the binding. After docking using Autodock Vina, one gets an output file. This output file allows one to view the interactions between the docked molecule, which I'll refer to as ligand from now on, and IL-18 when opened in PyMol. PyMol is a protein viewing software. To the right is something you might see in PyMol. This is actually the NSC61610 small molecule, shown in pink, bound to IL-18, which is shown in blue. From viewing the protein-ligand interactions in PyMol, I identified certain areas in the ligand or small molecule to add atoms or functional groups to improve binding. I drew the new molecules on Marvin Sketch and then docked them on IL-18 using Autodock Vina. I compared the binding energies given by Autodock Vina between the original NC61610 molecule and its analogs, and I also compared the drug viability outputs such as water solubility from a software called Swiss ADME. So actually coming up with the analogs involves a lot of trial and error. I had to view interactions between the small molecule and IL-18 in PyMol, and then predict where certain atoms could be added to make binding better. For example, adding a benzene group to make a molecule better fit in a certain pocket within IL-18. These were the analogs of IL-18 that were most promising. The first is the original molecule plus a cyclopentane circled in red, which is a ring of 5 carbons each bonded to 2 hydrogen atoms. The second is NSC6161 plus 1 carbon atom, just shown to the right, and circled in red. And this formed a methyl or CH3 group. So the results that I mostly compared between Autodock, Vina, and Swiss ADME were the structure, binding energy, and solubility. I already went over the structure, so now onto the binding energy. As predicted, the binding energy of the new compounds were lower than that of the binding energy of the original molecule. This indicates that these compounds will be more efficient at binding to IL-18, which is what we want. Second, the main parameter that I compared from Swiss ADME was solubility level. I didn't have the chance to calculate the solubility level for NS61610, which I'll do in future work, but based on the values calculated for the other two compounds, they have pure solubility, which means that they might need high dosages in order to achieve a therapeutic level, which is not ideal. So I'm not completely done with my project. I'm only in the initial phases of it. So in future work, I want to continue optimizing NS61610 and look at other Swiss ADME parameters as well as look at the solubility, which I didn't get the chance to do for the original molecule. I also want to optimize the other compounds proposed by the paper. And these compounds are NSC 80734 and NSC 201631. So thank you for listening to my presentation. I wanted to say a huge thank you to my mentor, Christine, and also Polygence for providing me the platform to conduct this research. Thank you.
Great job, Khushi. And so if you have any questions, feel free to put them in the chat or to put them in the Q&A. We did have someone request to share, so let's see if they have a question for you. Not sure if their screen is loading. One question that I have that I'm interested in that solubility piece that you were talking about, you had said kind of one of the trade-offs is that it would need to be in a really high dosage. Are there other ways to combat the pharmacology of that? Like, are there ways to improve its solubility outside of just increasing the dosage?
Yeah, so improving its solubility would actually be tackling the molecule structure itself. When the molecule is smaller, even though it's already a small molecule, when it has less atoms, it's more easily able to cross like the plasma membrane in cells. So if there's a certain structure or conformation that we could reduce these molecules to, then their solubility would greatly increase. And instead of providing such high dosages, which may not be ideal, we can lower the dosage and increase the therapeutic ability.
That's really cool. That makes a lot of sense. So are some of those future molecules that you're looking into, are some of them smaller and maybe have increased solubility?
Yeah, actually, I've tried optimizing some small molecules, which are actually like 10 atoms smaller than the ones that I showed. But I don't have data on that as of now, since I'm only in the initial parts of my project.
Okay, well, I'm on the edge of my seat waiting for that then, because that seems really awesome. And let me check. If anybody has any other questions for Khushi, again, feel free to put them in the chat. This was, we had a few students rescheduled to other sessions later in the day, so we have some time. If you have other questions in general, feel free to reach out, but otherwise I encourage you to pop around into other rooms where students are still presenting. We're so proud of all of the work that these students have presented. It is really amazing what they've been able to accomplish. And thank you all to our audience members for coming to support and hear our students present.